Hi, my name is Troy McEachron, Assistant Professor in the Department of Translational Genomics and Department of Pediatrics at the Keck School of Medicine of the University of Southern California. And today I'll be presenting some of the data that we obtained using NanoString profiling, and the title of the presentation was called Transcriptional Profiling of the Pediatric Osteosarcoma Microenvironment. So as an introduction to osteosarcoma, as this is a disease that not too terribly many people focus on, this is the most common malignant bone tumor in the pediatric and adolescent young adult age range. Pediatrics being defined as ages 0 through 14, and the adolescent young adult age range being defined as ages 15 through 39. So as we can see on the graph on the left, where rate per million is plotted on the y-axis and age of diagnosis is plotted on the x-axis, we can see that there is a peak during this adolescent young adult age range in both males and females, and on the right is a gross depiction of what an osteosarcoma would look like. These tumors do invade the bone marrow cavity as well, often cases. Osteosarcomas are genomically complex tumors with profound genomic instability, yet very few single nucleotide variants. So here we can actually see the rate of chromothripsis in osteosarcoma, which is a catastrophic genomic event where a specific genomic locus is actually shattered and then recombined in a random order, and that results in massive rearrangements of the affected locus. So this is just, on the left-hand side, this is whole genome sequencing that was performed from a patient with metastatic osteosarcoma, and the colored lines crisscrossing the inside of this Circos plot represent inter and intrachromosomal translocations, so massive amounts of structural abnormalities. On the right-hand side is data from a recent article, and just showing out of various pediatric tumors, solid tumors, CNS, and hematologic malignancies, osteosarcomas exhibit the highest number of structural variants and prevalent chromothripsis. Unfortunately, the survival rates for patients diagnosed with osteosarcoma have not improved over the last 30 years. Patients with metastatic disease do demonstrate an inferior outcome, as indicated by the survival plots underneath. On the left is the survival plot looking at patients with localized disease at diagnosis. On the right is the survival plot looking at patients with metastatic disease at diagnosis. The solid red line indicates overall survival, whereas the dotted blue line indicates event-free survival. And what we can see is that patients with metastatic disease at diagnosis have a relatively dismal prognosis. Data from different clinical trials shows that immune checkpoint blockade does not provide a therapeutic benefit to patients with advanced or metastatic disease. So then we asked a relatively simple question. Does the genomic complexity of osteosarcoma facilitate immune escape and or resistance to immunotherapy? So right around the time we were starting to ask this question, a paper came out and was very instrumental in helping us guide our thought process. So on the left, we can see that osteosarcomas have a variety of different copy number alterations, whether they be focal events or events encompassing chromosome arms or whole chromosomes. On the right, this paper demonstrates that focal copy number events have tend to have one outcome in sustaining proliferation. chromosome arm or entire chromosome copy number alterations can lead to sustained proliferation or escaping the immune system, and then the concept of neoantigen load as well can actually present targets, cancer-specific targets for T cells to recognize. So we know that osteosarcomas demonstrate aneuploidy, these focal and broad copy number alterations, and osteosarcomas do generate neoepitopes, albeit at a much lower level than that of most adult solid tumors, and the expression of these neoepitopes is very low. So putting this all together, we decided to ask the question, are there copy number alterations, whether they be focal, whole chromosome arms, or whole chromosomes, but are there copy number alterations in osteosarcoma that will impact the ability of these tumors to be recognized by the immune system? In doing so, we found that there are actually copy number gains of targetable immune checkpoints in subsets of patients with osteosarcoma. So here on the top, we can see Circos plots. So this is a means by which we can represent whole genome sequencing data. And on the left, the PDL1, PDL2 locus is amplified. In the middle, the B7H3 or CD276 locus is gained. And on the right, the locus encoding IDO1 is also amplified. The oncoprint on the bottom just shows how the distribution of these events, such that the PDL1, PDL2 locus is actually a shared locus, and there's tremendous overlap between them. The B7H3 locus and the IDO1 locus are different loci, so what we can see is that there are non-overlapping subsets of osteosarcomas that have copy number gains or amplifications of different targetable immune checkpoint genes. However, understanding or demonstrating that there are copy number alterations in targetable immune checkpoint genes in osteosarcoma is profound in that we were the first to discover this, and this provides a context for therapeutic intervention. However, these tumors do not grow in a vacuum. We have to understand the microenvironment that these tumors grow in. So how do these copy number alterations impact the tumor microenvironment? And at the same time, how does the tumor microenvironment impact the growth, progression, and clonal selection and expansion of these tumors? One thing I do want to note is the very interesting microenvironment of osteosarcoma. So these are indeed bone tumors, oftentimes which invade the bone marrow. The bone marrow is the site of postnatal hematopoiesis, so one can imagine that these osteosarcoma cells that invade the bone marrow are in the presence of various immature immune cells and progenitor cells. So this presents a very interesting type of microenvironment. So we wanted to ask the question, what does the microenvironment of osteosarcoma look like in primary disease, meaning tumors of the bone, and in metastatic disease, and specifically lung metastases? So to do that, we utilized a tissue bank at Children's Hospital of Los Angeles, and we retrieved pediatric osteosarcoma specimens. These specimens were formalin-fixed paraffin-embedded blocks. These were unmatched samples, so we have a subset of samples that were obtained from the lung, representing pulmonary metastases, and then another subset of samples that were obtained from the actual primary bone tumor. This chart demonstrates the sample. This chart demonstrates or characterizes the samples that were actually used for this study. So we can see age, sex, the site from which the tumor was derived or the specimen was derived, the pathological staging, and the disease status. So these tumors are indeed bone tumors, which means that they are heavily calcified. So in order to actually cut sections from these blocks, we had the blocks acid decalcified. Both the metastatic and non-metastatic specimens were acid decalcified, such that there was no batch effects due to one set of specimens being decalcified and the others not being decalcified. So the tumor specimens were cut into serial sections. A portion of those specimens were scraped, and the RNA was extracted from these samples using the Agincourt Forma Pure RNA kit. The RNA from these samples was then utilized for the gene expression profiling using the NanoString pan-cancer IO360 panel. The remaining sections were used for tissue staining, namely hematoxylin eosin and immunohistochemical analysis. The IHC, H&E, and gene expression data was combined, analyzed, and integrated and interpreted such that we can really understand what is going on at the gene expression level and the protein level in the tumor microenvironment of metastatic and non-metastatic osteosarcoma. One thing I do want to note is that the specimens used for this study came from patients who responded poorly to chemotherapy. This is important because in the clinical setting, patients with osteosarcoma that would have received any kind of immunotherapy would have already failed the standard of care chemotherapy. So we wanted to ensure that we were looking and investigating the tumor microenvironment in the right context of patients who have failed prior chemotherapy regimens. We did assess tumor viability prior to experimentation to ensure that we weren't utilizing specimens that had extensive necrosis. So all of the specimens that were used had sufficient viable tumor to go on for downstream analysis. Given the fact that the IO360 panel is really focused on a hybrid capture approach of capturing mRNA for subsequent counting, we needed to ensure that the RNA that we had was of sufficient quality. This is extremely important given the fact that all of the samples were acid decalcified, and acid will heavily degrade nucleic acids. So what we can see here is that for all of the specimens, we had sufficient concentration of nucleic acid, and these nucleic acid species were 300 nucleotides in length or greater, as indicated by the %DV300 value. So this gave us confidence that the nucleic acids that were extracted were sufficient for going into the NanoString IO360 profiling. On the right is a trace from the Agilent tape station 4200, just showing what the distribution of nucleic acid molecules actually looks like. At the bottom, it is the nucleotide size and length, so we can see that there is a sufficient number of molecules that RNA molecules that is of sufficient size. So now I'll present the results from this NanoString profiling. So the first thing that we wanted to do was to get an overall view of what the profiles look like between metastatic and non-metastatic osteosarcomas. So we performed an unsupervised hierarchical clustering, and what we can see is that this clustering separates the metastatic and non-metastatic osteosarcoma specimens into two distinct and non-overlapping clusters, whereas the metastatic osteosarcoma specimens are labeled in gray at the bar on the top, and the non-metastatic osteosarcoma specimens are labeled in black, and again, the bar on the top. This color convention, gray indicating metastatic and black indicating non-metastatic osteosarcomas is consistent throughout this presentation. Next, we looked at the expression levels of various inhibitory and stimulatory immune checkpoints. And for the most part, what we can see is that the non-metastatic specimens have higher expression of various immune checkpoints in comparison to the metastatic specimens. There are three notable exceptions, one of them being HVEM or TNF-RSF14, in which the metastatic specimens have higher expression than the non-metastatic specimens. The same can be said for PDL1 in the bottom left-hand corner of the panel, and as well as for VISTA, which is the bottom row, second from the right. So with the exception of these three checkpoints, the expression of various immune checkpoints is greater in the non-metastatic specimens versus the metastatic specimens. We then looked at a variety of functional metagene signatures, and this is one of the strengths of the assay, in which there are a collection of functional metagene signatures that describe different processes within the tumor and the tumor microenvironment. In this heat map, the functional metagene signatures are on the left, and on the right-hand side of the heat map are the genes and gene signatures that comprise each of the functional metagene signatures. Again, unsupervised hierarchical clustering separates the metastatic and non-metastatic osteosarcoma specimens into two distinct clusters that do not overlap. We then wanted to look at just be able to apply statistics and see if there were any statistically significant differences between these different functional metagene signatures and in metastatic and non-metastatic osteosarcomas. And what we can see is that the metastatic specimens demonstrate a decreased enrichment in these functional gene signatures when compared to non-metastatic specimens. I do want to point your attention to the bottom right-hand side of the panel, where we're looking at immune cell population abundance. This was one of the most interesting findings that we observed, and this is what we decided to follow. So we took those differences in immune cell population abundance and started to look at the different immune cell subsets that are described in the NanoString panel, and looking at their relative abundance between metastatic and non-metastatic osteosarcoma specimens. And what we can see here, with the exception of mast cells, the metastatic osteosarcoma specimens, again in gray, contain fewer immune cells than the non-metastatic osteosarcoma specimens. This was incredibly important, as this concept of lymphocyte exclusion has gained a lot of traction recently, where there may indeed be immune cells present. However, the localization of the immune cells is such that the cells do not actually penetrate the tumor, as opposed to being confined to the periphery of the lesions, or not present at all. So in order to visualize the distribution and localization of T cells, we utilized immunohistochemistry. We specifically stained for CD3 epsilon to indiscriminately visualize all T cell subsets. So in this instance, we were more interested in understanding we were more interested in being able to visualize all T cell subsets without respect to whether these are CD4, CD8, Th1, Th2, Treg, so on and so forth. The top row is hematoxin and eosin staining for the sake of demonstrating that the regions that we were looking at did indeed contain viable tumor. The middle row shows CD3 epsilon immunoreactivity, whereas on the left we're looking at the metastatic osteosarcoma specimen, specifically on the periphery of the tumor, adjacent, so the normal lung parenchyma butting up against the actual metastatic lesion. The middle panel is an image from the core of that same metastatic lesion. On the right, we are looking at the core of a non-metastatic osteosarcoma. The boxes in each one of these panels is just a region of interest that is further magnified in the bottom row. So what we can see is that in the metastatic osteosarcoma specimens, CD3 immunoreactivity is confined to the periphery of the specimen, and we do not see CD3 immunoreactivity in the core of the specimen. However, in the non-metastatic osteosarcomas, we do indeed see CD3 immunoreactivity distributed throughout the core of the tumor. On the right-hand side, we quantified this immunoreactivity, and what we can see is that the non-metastatic specimens express higher levels of CD3 immunoreactivity, and this is consistent with the NanoString gene expression profiling data. I do want to note that although the non-metastatic specimens have more T cells that infiltrate the tumor, the abundance of these tumor-infiltrating T cells is still relatively low in comparison to a variety of other solid tumors that, such as lung cancer, so on and so forth. Now we wanted to look at the activation status of some of these T cells. So we've already seen that 4-1BB expression is lower in the metastatic versus non-metastatic specimens, and we can see that here on the bottom right-hand side. We can see that 4-1BB expression in the metastatic specimens, which is indicated in gray, is lower than that of the non-metastatic specimens indicated in black. And we then did a correlation analysis to get an idea of, does the expression of 4-1BB correlate with the abundance of a variety of lymphocytes. Now, we chose 4-1BB because this is an activation-induced co-stimulatory molecule that serves as a surrogate for lymphocyte and NK cell activation. So in doing this correlation analysis, what we can see is that in the metastatic osteosarcoma specimens, we do not see a significant correlation between 4-1BB expression and the lymphocyte and NK cell populations listed. However, in the non-metastatic osteosarcoma specimens, we do see very, very, very strong correlations indicative of activation status of these lymphocyte populations. Going back to this concept of lymphocyte exclusion, our data shows that in the metastatic osteosarcomas, the T cells are actually confined to the periphery. And when we start looking at this a little bit further, one of the things we realize is that angiogenesis can also be a key mediator to lymphocyte exclusion, whereas the phenotype and functionality of the tumor vasculature can actively prevent T cells from either extravasating or render the T cells that do extravasate inactive due to a variety of different immune checkpoints expressed on the surface of the endothelial cells. We can see B7H3. Also, it's been shown that IDO1 and PDL1 have also been expressed on the vasculature, the endothelial cell surface of tumor vasculature. So the first thing we wanted to do is to look at the endothelial cells using the NanoString data. So we chose E-selectin to as a surrogate marker for activation. E-selectin is expressed exclusively in endothelial cells, and it's transcriptionally upregulated in response to inflammation. So this serves as a marker of an activated and functional endothelium. There are a variety of other markers, such as ICAM1 and PCAM1, that people often use. However, both ICAM1 and PCAM1 can actually be expressed in both endothelial cells and tumor cells, whereas E-selectin, encoded by cell E, is specific to endothelial cells. So what we see is that the metastatic osteosarcoma specimens express lower levels of E-selectin in comparison to the non-metastatic osteosarcoma specimens in black. We wanted to assess the endothelial cell abundance to determine if the differences in E-selectin was due to a decrease in tumor vascularity. So in assessing the endothelial cell abundance between metastatic and non-metastatic osteosarcomas, what we see is that there is no significant difference between metastatic and non-metastatic osteos, suggesting that the E-selectin, or the differences in E-selectin between the two groups of specimens, is not due to a decrease or increase in endothelial cells. We performed a Pearson correlation analysis between the endothelial cell abundance and E-selectin expression in both metastatic and non-metastatic specimens. On the bottom left, we can see that there is a strong correlation between endothelial cell abundance and E-selectin expression in the non-metastatic osteosarcomas, whereas on the bottom right, this same analysis reveals that there is not a strong correlation between endothelial cell abundance and E-selectin expression in the metastatic osteosarcoma specimens, suggesting that these endothelial cells in the metastatic tumors are hypo-responsive. Next, we wanted to investigate vascular instability in the context of metastatic and non-metastatic disease. Angiopoietin 1 and angiopoietin 2 opposingly regulate vessel stabilization and angiogenesis, such that expression of Ang1 facilitates parasite coverage, vessel maturity, and vascular stabilization, whereas expression of angiopoietin 2 induces parasite detachment and vascular instability. And this is what the diagram indicates. Ang1 is an agonist of the Tie2 receptor, whereas Ang2 is an antagonist of the Tie2 receptor. So these two molecules, the balance of these two molecules in binding to Tie2 dictates to a large degree the stability of the vasculature. We queried Ang1 and Ang2 expression in metastatic and non-metastatic osteosarcoma specimens, and what we can see is that there is a decrease in Ang1 in metastatic osteosarcoma specimens in gray in comparison to non-metastatic osteosarcoma specimens. There are no differences between Ang2 expression in metastatic and non-metastatic osteosarcomas. We then calculated the Ang2 to Ang1 ratio to serve as a surrogate for vessel stabilization, where an increased ratio favors vascular destabilization, while a decreased ratio favors parasite coverage, vessel stability, and vessel maturation. What we can see here is that the metastatic osteosarcoma specimens indicated in gray have an increased Ang2 to Ang1 ratio in comparison to that of the non-metastatic osteosarcoma specimens. And what this shows is that, or what this indicates or suggests, is that the vessels in the metastatic specimens are unstable. We then looked at the ratio, the Ang2 to Ang1 ratio, as a surrogate for vascular instability, and correlated this with lymphocyte abundance in metastatic and non-metastatic osteosarcoma. So what we can see is that in the context of metastatic disease, the Ang2 to Ang1 ratio inversely correlates with inversely correlates with lymphocyte abundance. These correlations don't, these significant correlations do not exist in myeloid and granulocytic lineage cells. In the non-metastatic osteosarcomas, we do not observe inverse or positive correlations between the Ang2-Ang1 ratio and lymphocyte or immune cell abundance, suggesting that the metastatic osteosarcoma specimens are actively associated with lymphocyte exclusion, and that this correlates with vascular instability as assessed by the Ang2 to Ang1 ratio. We also looked at VEGF expression in metastatic and non-metastatic osteosarcoma. Pathophysiological levels of VEGF can induce endothelial cell dysfunction and is associated with vascular instability and resistance to anti-PD-1 therapy. So on the top row, what we can see is VEGF-A immunoreactivity in 3 different metastatic osteosarcomas, and on the bottom row illustrates VEGF-A expression in 3 different non-metastatic osteosarcomas. So we can see clearly that the metastatic osteosarcoma specimens express higher levels of VEGF than the non-metastatic osteosarcoma specimens. And this is quantified on the right. On the top right, it's quantification of the VEGF-A immunohistochemistry. On the bottom right is quantification of VEGF-A gene expression using the NanoString profiling. And what we can see is that the trends are the same, where metastatic osteosarcomas express higher protein and mRNA levels of VEGF-A in comparison to non-metastatic specimens. And in both cases, these results are statistically significant. Similar to what we did before with the Ang2 and Ang1 ratio, we decided to perform correlation analyses between VEGF and lymphocyte abundance. We see very, very similar results to what we saw previously with the Ang2 to Ang1 ratio comparison, where VEGF inversely correlates with lymphocyte abundance in the context of metastatic disease, whereas in non-metastatic disease, these correlations do not exist. The next thing that we actually wanted to look at was the tumor inflammation signature. So the tumor inflammation signature is a weighted metagene signature that measures the degree to which the pre-existing anti-tumor adaptive immune response is suppressed. Given the fact that we see decreased abundance of various immune cells in the metastatic osteosarcoma specimens, we were expecting that the tumor inflammation signature would actually demonstrate that. First thing that we did was to look at the tumor inflammation signature across various pediatric malignancies just to gain context as to what the tumor inflammation signature in osteosarcoma looks like in comparison to various other tumors. Starting from the left, we have retinoblastoma, medulloblastoma, adrenocortical tumors, Wilms tumors, ependymomas, high-grade gliomas, choroid plexus carcinomas, T-cell acute lymphoblastic leukemias, low-grade gliomas, rhabdomyosarcomas, mixed lineage leukemias, neuroblastoma, B-cell acute lymphoblastic leukemia, osteosarcoma, acute myeloid leukemia, and melanoma. And what we can see is that osteosarcoma actually has a relatively high tumor inflammation signature in comparison to various other pediatric tumors. This data was obtained using the St. Jude dataset. When we then look at the target osteosarcoma dataset and separate specimens into metastatic and non-metastatic, what we see is that the tumor inflammation score does not differ between metastatic and non-metastatic specimens. We did the same analysis with the specimens used in our study, and we see the same results, that the metastatic and non-metastatic specimens do not differ as when it comes to the tumor inflammation signature or the tumor inflammation score. From this, we were left with a hypothesis that we're currently trying to validate, such that there are differential roles in the tumor vasculature in the context of metastatic and non-metastatic osteosarcoma. So on the left, what we hypothesize is going on in non-metastatic specimens is that lymphocytes are actually able to extravasate, therefore they are able to leave the vasculature and infiltrate the tumor. And this is due to an increased expression of adhesion molecules and a much more balanced ratio between VEGF, Ang1, and Ang2. However, in the context of metastatic osteosarcoma, the lymphocytes are actually excluded, and this is mediated by a decrease in adhesion molecules and an imbalance between Ang2 and Ang1 and an increase of VEGF signaling. The conclusions from the study are the following. First, non-metastatic osteosarcoma specimens demonstrate increased gene expression levels of several immunotherapeutic targets, whether they be agonistic or inhibitory. Metastatic osteosarcomas contain fewer immune cells than the non-metastatic specimens, and that T cells are actually excluded from infiltrating the metastatic foci and are likely hypo-responsive or inactive. The vasculature in metastatic disease exhibits signs of dysfunction, and this inversely correlates with lymphocyte abundance in the context of metastatic osteosarcoma. And interestingly, the tumor inflammation score does not differ between metastatic and non-metastatic osteosarcoma specimens. The implications for our research are the following. First, vascular dysfunction, and we assess that using the Ang2-Ang1 ratio and expression of E-selectin. Vascular dysfunction actively promotes lymphocyte exclusion in metastatic disease. In the context of inflammation, such as that of malignancy, excessive VEGF signaling can actually induce endothelial cell anergy, and this is a state in which the endothelial cells are hyporesponsive and do not respond well to inflammatory stimuli and redistribute the adhesion molecules and or downregulate selective adhesion molecules, the end result being that lymphocytes are not able to bind the luminal surface of the vasculature and extravasate. So we feel that we have evidence to suggest that this actually may be occurring in metastatic disease. Vascular normalization has become incredibly popular lately in terms of targeting VEGF and Ang2 signaling, and this transient vascular normalization can actually increase lymphocyte infiltration, and there are several trials right now that are combining anti-angiogenics and immune checkpoints to see if the vasculature can indeed be stabilized to allow for immune cells to leave the vasculature, infiltrate the tumors, and then relieve that inhibition using checkpoints. The tumor inflammation signature may not accurately reflect the biology of pediatric osteosarcoma, and the reason that we say this is that osteosarcomas are a pediatric malignancy of mesenchymal origin, and the tumor inflammation score was actually derived from adult carcinomas. So these differences may actually reflect the fact that the tumor inflammation score may be better suited towards specific types of malignancies and not well suited towards pediatric mesenchymal-based malignancies such as that of osteosarcoma. So I thank you for your time, and if you have any questions, please feel free to send an email, and I will respond to these questions at my earliest convenience. Thank you.